Hey, hi everyone. Today we are going to have our last lecture of new material that you will be responsible for this upcoming Friday's exam one. So hopefully all of you are aware that our first exam is this Friday. And so it'll cover all the stuff that we've talked about so far in the semester up through and including today's material. Because today we're going to wrap up our discussion about our introduction to how T cells work, in particular the T cell receptor. And then we'll start after the exam looking at a closer appreciation of how B cells and T cells actually develop. So let's make sure that we're all aware. Here we are. So today is the last lecture on chapter 5. So after today's lecture, there will be a quiz made available in Laulima, and that quiz will help you, get familiar with the chapter 5 material. And so that will be due the normal time, Thursday night at 11:55 p.m. So today we'll have our last lecture on chapter 5. Wednesday, when we meet for class, it will be a review session. We'll go over some of the important topics for the exam, hopefully put you in a good position to be able to wrap up your studying, and then Friday is the exam. So this week's quiz, I'll make it available today, and I will set it up so that when you submit your quiz, you will immediately get your score and also the feedback so that you can see the answer key. Because after all, the point is I want you to be able to use this to study, right? So that means, different of you will take it at different times. Some of you will just go ahead and take it today. Others of you will wait until Thursday. So what I ask is, because those of you who are finishing earlier, you'll have access to the correct answers, please do not share the quiz answer key with your classmates who haven't yet taken the quiz. Because even though you might think you're doing them a favor, you're really not, right? Because if they're not able to try the quiz on their own and actually think about these questions, that's actually hurting their ability to use the quiz as a study tool. All right, so keep that in mind. In terms of the format of the exam will be 50 questions, multiple choice, taken over Laulima. And so I will send an announcement today with some more details, but briefly, you do not have to log into Zoom that day. It's all taken on Laulima. So you would just go ahead and at class time, so at 12:30, log on to Laulima. The test will be available, and then you just go ahead and you take your test. Remember, the test is closed book, closed notes, closed internet, and you have to take it by yourself. All right. So as a result of the whole online teaching situation, there are certain restrictions and guidelines about how the test is set up, right? The time is pretty limited. You have 50 minutes to answer the 50 multiple choice questions. If you think about it, that's no different than any normal semester, right? Because the class period is 50 minutes. But the thing is, the questions will be set up so that as you answer a question and move on to the next one, you will not be allowed to go back and change the response that you previously put. And I know many of you, would probably hate that because you said, but that's not like a normal test packet, right? Where you're given a set of pages and you can flip back and forth and jump around and take them in any order that you want. Unfortunately, the truth is that taking the tests online this semester, this is not a normal set of circumstances, right? And so in order to make sure that everybody takes the exam in a fair, equitable way, because I have no control over what you guys are going to be, where you're going to be set up, and I won't be able to see you at all. You're not going to be proctored, right? I've set it up this way to keep it the same for everyone. So in other words, there's not enough time for you to read a question and try to Google the answer or frantically flip through your notes, right? Because you're going to run out of time if you try to do that. And likewise, right, because you can only answer the questions in order, this is to discourage students from just getting on a conference call on their cell phone with several of you taking the test together, or comparing, what you got or how you answered certain questions, and then being able to go back and change your answers to match what your friend put, right? So I know that sounds kind of strict, but hopefully you all understand that that's why we're doing it this way, right? To keep it fair. As I always tell the students, if you're honest, as almost all of you are, right? Then if you study hard, right, and you earn a certain score on this exam, I always say, how would you feel if you found out that like a handful of other classmates all cheated by taking it together, right? And they got 100% as a result, and their exam score counts as much towards their grade as yours does towards yours, right? So that's why, to keep it fair, we have these rules. So again, those details you'll see in an announcement that I'll send out later today, just so that you know what to expect. Also importantly, for students who make use of the COCUA services, COCUA does send me a list of students who are in their database who have requested COCUA services this semester. But it's up to you to make sure you're on that list, right? Because if you're on that list, I'm not going to know to set you up with any kind of special time accommodations, right? And importantly, right, if you email me and say, oh, you want COCUA time accommodations, if you're not on the list that COCUA sent me, I cannot give you those accommodations. Again, to keep it fair. All right. So if you need COCUA accommodations, make sure that you've contacted COCUA, or you can email me and ask me if your name appears on the list. All right, so it's up to you to take care of yourself and make sure that I have that important information. All right, so we can talk more about the exam on Wednesday, but I wanted to go ahead and give you a heads up that that's what it's going to be like. All right. So let's finish talking about T cell receptor and some of the important things about T cell receptor. So here's a photo of one of my kind of favorite places in town. Many of you might recognize it, right? This is the food court at the Shirokiya food court at Ala Moana. All right, so this picture was taken, I don't know, like maybe like 3 years ago or 4 years ago, when it was like this really bustling kind of place. I mean, obviously now these days, I don't even think they're open because of the pandemic, but even leading up to the pandemic, things started getting kind of sad there. There used to be like 100 different vendors there, and it was kind of a fun place to wander around, finding all kinds of good stuff to eat. But the problem is, if you have 8 different ramen places, then eventually, right, 7 of them are going to close down. But anyhow, so many of you have probably been to the Shirokiya Village Walk food court, and you wander around looking for something good to eat, right? And as you're wandering up and down, you see things like, oh, the different vendors, they have like nice colorful displays, letting you see what's going on in there, right? Or they might even have some of their food set out so you can see it, right? And if you think about it, right, this is what they have to do, because it's not like when you're wandering up and down the aisles looking at visiting the different vendors' booths that you can just let yourself into their little kitchen area and actually peek at what they're cooking, right? You're limited to being outside of their booth, right? So in other words, you rely upon them to advertise what their food is like, what they're making, to give you a chance to kind of shop around and see if you can find something that you like. Whether it's some nice sushi, or these are my daughter's favorite super fancy musubis, right? So this is from the little musubi cafe that I think they still have there. These are those nice musubis that are like spam, egg, bacon, and avocado, right? And the amazing thing is, when they first opened, these were like $2.75 or something like that. Okay, so why do I mention this? Okay, I mention this because I think we can all relate to what's happening here, right? Your ability to find what you want and what you're interested in is entirely dependent upon the food vendor, the restaurateur here, advertising and showing you what's going on inside their particular kiosk, right? So that's like the way our body's cells have to advertise to our immune system what is going on inside of them. This semester, when we first started talking about the immune system, we used pretty straightforward examples of things like bacteria that get into your body from a cut, right? And these bacteria then, if you think about it, if they're inside your tissue, infecting your tissue, they, many times are still outside of your individual cells. They're just in the spaces between your cells, right? In the fluid, in the sort of interconnecting tissue. And as a result, components of your innate immune system, for example, like the complement proteins, or even cells like macrophages, dendritic cells, things like that, right? They're just patrolling around, again, between your cells, right? So that's fine if the pathogen is limited to being outside of your cells. But what that kind of means is that those immediate effector cells and components like the complement proteins, they're really just concerned with what's kind of in between your cells, right? They don't have the ability to actually see what's going on inside of your cells, right? I mean, that kind of makes sense. You've already learned enough about complement proteins to know that they, are limited to kind of acting on the outside of a cell. They don't go into a cell and kind of try to cause any kind of action there. Likewise, a macrophage is not going to somehow go into one of your cells and check things out, right? It's stuck to being on the outside. That's why we evolved things like our interferon response. So when viruses infect our cells, right, a lot of times our cells send out different types of interferon cytokine messages to alert our immune system. But something that's even more sort of fundamental and basic is simply the fact that our body's cells always advertise on the outside of their cell membrane what's going on inside of them, right? And how do they do that? Well, it's not that much different than having nice colorful posters of sushi, or more specifically, actually having the little musubi rolls out for customers who are coming by to actually see, right, and take. Of course, what we're talking about here is the fact that our cells present small peptides from proteins that are coming from inside of them out on their surface on these special MHC platforms. Right? This is what we've been discussing for the last couple lectures. So let's remember here, right? All of our cells in our body, at least all of the cells that have nuclei, right? So red blood cells are kind of an exception for a lot of things, because mature red blood cells are nothing really more than just sacs of some protein, including lots of hemoglobin. So they don't have a lot of MHC on their surface. Most of our body cell types express a lot of MHC, right? But remember, there's two types of MHC. And depending upon which MHC type a particular cell is using to advertise or display a small piece of protein that came from inside of it, right, makes all the difference in the world as to what your immune system does in terms of interpreting that. Remember, a subclass of our cells express what we call MHC class 2. So these are mainly limited to cells of our immune system whose job it is to pass messages on to other immune cells. So things like our dendritic cells that are all about collecting different bits of evidence of things like pathogens or little bits and pieces of things and showing them off on the outside, or things like B cells that have immunoglobulin on their surface. And if things are attaching to their immunoglobulin, right, the immunoglobulins, if they're still bound on the surface of a B cell as a B cell receptor, these are constantly being recycled. Right? I mentioned to you last time that cells are always recycling the stuff that's on their membranes, bringing proteins back in, breaking them down, right, replacing them with new versions of it. So things that are bound to immunoglobulins on the outside of a B cell get brought in, and the B cell then has the opportunity to present those things, right, on MHC. And macrophages, our friends, the macrophages, right? Great example of something that its job is to go around and eat things up and then display things, right, for other immune cells to see, right? So all of those examples, the dendritic cells, the B cells, the macrophages, right, as part of doing their job, they present peptides that they've taken in as part of doing their job on MHC2, right? So not all cells in our body have MHC2 on their surface, only these special cells that need that as part of their normal cellular job as being part of the immune system have MHC2. So let's look at this cartoon. So this is kind of a review, Right. Here we have, let's say, a macrophage, this big gray cell, and it's been eating up things like, let's say, for example, here, this red oval is some sort of antigen, some sort of molecule that came off of a pathogen, a bacteria, virus, whatever. Remember, the macrophage will phagocytose it. It'll have it in this endocytic vesicle, and then the endocytic vesicle will fuse with something like a lysosome, and that will form the phagolysosome, where that bacterial protein gets broken down into small little red dots. And remember, what are the red dots? The red dots are small fragments of proteins. In other words, small little peptides, right, that are maybe on the order of 8, 10, 15 amino acids in size, right? So the ones that fit, because they're that size, can be loaded on MHC2. So the MHC2, where was that made? Well, it's just a protein, just like any other protein in your body, right? So in this case, like many proteins, it finishes being put together in the endoplasmic reticulum. And then remember, we learned last time, at this point, it's kind of capped on top of it so that no small peptides from inside the ER can be loaded on it, right? So in that unloaded form, the MHC2 is sent through the Golgi to be packaged up in a little vesicle, and that vesicle then fuses with the phagolysosome, and that's when MHC2 is loaded up by the small little peptide fragments, and then it goes to the surface and now displays this little red dot, this peptide, to the outside, right? So remember, that situation is this macrophage just doing its job, right? Is the macrophage infected? No, not in this case. Is it damaged somehow? No, right? It's actually doing its job the way it's supposed to. It's eating things in and showing them on MHC2. The contrast would be, for example, if indeed this macrophage did get infected, right? Because macrophages, just like any other cell in the body, can be infected by things like virus, for example, then you would find the antigen present in the cytoplasm, right? So these red hibiscus represent pathogenic proteins that are floating around inside the macrophage's cytoplasm because they got in there as the result of infection, right? Remember, we learned that these proteins will get unfolded and fed through the recycling bin of the cell, the proteasome, where it will be chopped down into small little red dots, into small peptides. And then those peptides will go through various channels in the endoplasmic reticulum. And that's where they will be loaded on MHC1, right? So MHC1 protein is also being put together here. Remember, the way the cartoons draw it is MHC1 looks different than MHC2 because MHC2 is two chains, an alpha and a beta together. The MHC1 is one long alpha chain, and then you have this funny globular beta microglobulin as part of the structure, right? When the MHC1 is put together in the ER, pretty much right away, it can get loaded up with peptide, right? Maybe there are other enzymes here, like ERAP, that help trim these little peptides down to the right size to optimally fit in MHC1, and then that MHC1 moves through the Golgi. It's packaged up, already loaded with its peptide on it, and it goes to its surface, right? So the important thing is whether this red dot is displayed on MHC1 versus MHC2 will determine what kind of T cell that comes along and bumps into this macrophage will actually activate, right? And that then will determine the fate of this macrophage. Right? On the left side, it would be, hopefully you guys know, right? It's waiting for a helper T cell, a CD4 positive T cell that would recognize this and maybe encourage the macrophage to really increase its activity, right? Its way of telling the macrophage, you're doing an awesome job, keep it up. In fact, we need you to do even more, right? On the other side, right, maybe a cytotoxic CD8 T cell would come along and recognize this, right? And then in that case, it would say, oh, macrophage, you're infected here by virus. Sorry, you need to go ahead and take yourself out of the picture here. And it would induce this macrophage to undergo apoptosis. So remember, the ability of a cytotoxic T cell or a helper T cell, so the purple T cell here or the blue T cell, to be able to recognize the same red dot that's being shown to it by another cell is dependent upon two things, right? One is by chance it has to have the right T cell receptor, so the right alpha beta T cell receptor that was put together through VDJ recombination when this T cell was first developing. And here in this by chance, this purple T cell over here has an alpha beta T cell receptor that recognizes the red dot in the context of it being presented on MHC, right? But the interaction here between the T cell receptor and the antigen on MHC, there's not enough information right at this junction point for this T cell to know whether or not this is actually MHC1 or MHC2, right? So how does it know that this is MHC1? It knows because on its surface, it also has CD8 co-receptor, right? This purple set of receptors here, alpha beta. And so that then swings over, and that's the right shape to bind to MHC1. And therefore, the combination of the T cell receptor activating plus the CD8 co-receptor then tells the CD8 T cell, yeah, this is an infected cell, infected with whatever this foreign pathogen peptide is that this T cell was kind of chosen to recognize, right? And then cytotoxic messages would be sent to this target cell to kill it. Okay? On the other hand, CD4 helper T cell comes along. It might have a T cell receptor that is almost indistinguishable than the one on the left over here. So it also recognizes the same red dot on the surface of MHC, right? But it has CD4 co-receptor. So the CD4 co-receptor, when it swings around, it says, yes, this is MHC2, right, that it's bound to. And that then will stimulate the helper T cell to do its helping job. It will send cytokine messages that encourage the antigen presenting cell, like the macrophage, right? Hey, you're doing a good job. Keep it up. Do even more. Right. So importantly, let's talk about this. Where does that red dot come from? So that came from originally the pathogen. So here's bad clown. So bad clown's infecting the tissue here. Right? If bad clown is taken up by one of these yellow cells up here, for example, if bad clown is infecting this target cell, right, he will then be broken down in the cytoplasm, bits and pieces. So these colored dots are all just bits and pieces of bad clown, different small, short peptide sequences, right? So in this case, the red dot comes from infection in the cytoplasm. Over here, bad clown would have gotten into the antigen presenting cell through being eaten by the macrophage, right? So very different scenario. And then the macrophage would load up its MHC2 with the red dot. So I know the reason I go over this over and over again, because this is one of the most important sort of sets of concepts in understanding T cell activation, right? You need to be able to understand this concept of what's going on here. Because it's the T cell receptor, the TCR, that is the thing that's the right shape to recognize, for example, that this is bad clown, right? Whereas the co-receptor, whether CD4 or CD8 is present and binds there, this is what's going to then interpret that this is the correct signal for this particular T cell to activate. One of the things that students always ask me, and a couple of you have always asked me this, right? How you say, like on the left, why is it only CD8 T cells would even come and contact this cell, right? Or why is it on the right that only these light blue CD4 helper T cells would come and contact the cell, right? That's what kind of messes you up because you don't understand that. No, the point is, right, there's an equal chance at any given time that there would be CD8 cytotoxic T cells pressing up against this antigen presenting cell on the right. And there'd be an equal chance, right, of helper T cells pressing up against this infected target cell on the left. But the point is, if that were to happen, or when that happens, right, even if the T cells see the red dot that their T cell receptors like, because they don't have the ability, or their co-receptors don't pick up on the appropriate MHC type, right? That's what keeps this from moving on to the next step of the T cell activating, right? So therefore, the CD4 helper cell pressed up against this target cell on the left, it won't activate, right? Because its CD4 co-receptor doesn't bind to MHC1. It only binds to MHC2. And you think like, well, why is that important? Because think about this, right? If the CD4 helper T cell activated against this target cell and started helping this infected target cell do what? Maybe make more virus particles, right? You don't want that, right? Likewise, the other way is if this cytotoxic T cell pressed up against this macrophage here that was presenting the red dot, and then this cytotoxic T cell activated on the right over here, you don't want that either, right? Because then it would kill this macrophage, right? So that's classic killing the messenger. The macrophage is just trying to do its job, right? So importantly, that's why this idea of the driver's licenses, to me at least, makes intuitive sense, right? You could have the exact same ID being shown on these two different driver's licenses, but one on the left is your full-blown normal, like full privileged Hawaii driver's license that can be used, for driving around on your own, whatever. The one on the right is maybe your limited purpose driver's license. Maybe this is the one that you use when you're like 15 or 16 and first learning how to drive, and you have to have a grown-up with you or something, right? And that's why the information that's carried by this other driver's license says not acceptable for official federal purposes or something like that on it, right? Okay, so therefore the driver's license itself contains information on how to interpret the identity of the picture here. All right, so let's move on and talk about then how it is that if a protein that comes from a pathogen like bad clown, or any protein for that matter, right? Because remember, I've told you already, if you think about it, at any given moment, like right now when you're sitting here, the majority of your cells in your body, in fact, maybe even all of them, right? There's no sign of pathogen, right? If you don't have an infection going on. So what are these cells in your body displaying on the outside? They're just displaying regular bits of normal peptides that come from normal, everyday, healthy proteins that are coming from inside of them, right? Again, your cell advertising to your immune system what's going on inside of it is just as much about assuring your immune system that everything is normal inside of the cell as much as it is about alerting your immune system that something weird is going on inside of your cell, right? Because as T cells are bumping into cells as they're going around in your body, think about it. The majority of cells that they're going to synapse with, bind to, and check out what's being presented to them are normal, healthy body cells, right? So those normal, healthy body cells are displaying normal, healthy fragments of proteins that are just being recycled inside of them. Right? If there's an infection, then the presence of little peptides that come from the pathogen are simply among the different things that are presented on the outside of the cell, right? So that if T cells come along and recognize those little signals, right, they will get excited. So let's look at this here. Let's pretend a pathogen like bad clown gets into a cell, either through infection or through the cell phagocytosing eating. Right? So bad clown, he's a pretty complicated dude, right? So you can think about it as a pretty complicated set of proteins. So if he gets broken down into all these little fragments, these short peptides, you can think about it as these different peptides representing different types of bad clown features, right? Maybe it turns out the red dot represents his straw hat. The green dot, small little peptide, represents his eye makeup, stuff like that, and the blue represents something like, I don't know, his beard or something like that, right? So these are all different fragments of amino acids, and they might as well be like completely random, because think about it, right? If you think about how many tens, if not hundreds of thousands of different proteins you have floating around in your body right now, even if you have like another protein that comes from a pathogen, if you take that protein apart into random little fragments, the actual composition of those fragments, right, it might as well be infinite possibilities, right? So how is it that our MHC platforms on the surfaces of our cells can handle accommodating, holding on, and presenting such a huge, possibly infinite number of different amino acid pieces, fragments, right, short peptides? Right? So if you look here, right, remember this is what the MHC looks like if you're staring straight down at it. Right? So on the left here, this is just showing the different colored dots that represent bits and pieces of bad clown. And in this case, the red dot, we'll just say for fun, hey, this is the straw hat part of him, right, gets presented on this MHC1. So here it is here, right, and how we normally see it. Now let's stare straight down from the top. So when you stare straight down at the top, you see the cleft, right, that's holding the peptide. And that cleft is made of these two sides, this left side that comes up, the right side that comes up, all part of that one long chain that makes up MHC1. So these are the two sides of the cleft. And here's the red dot, right? This is the short amino acid peptide. I mean, you can kind of, I don't know, let's count. You can see little side chains sticking out. 1, 2, 3, 4, 5, 6, 7, 8. Oh, okay, maybe this is like 8 amino acids strung together. But again, this might as well be one of an infinite number of possible different little shapes in here, right? So how can MHC actually do an equal job of hanging on to all of these? The point is, it can't, right? One specific MHC platform, right, with one specific shape will be able to hold on to some short amino acid sequences better than others, right? And some amino acid sequences, it might, quite frankly, be completely lousy at hanging on to. And those little fragments, right, wouldn't be displayed on the outside of the cell. So think about it this way. Any given cell in your body not only displays MHC1 on it, but it's not just one type of MHC1. Any given cell in your body has several different variations of MHC1 platforms on there. They're all MHC1, right? Meaning they're all meant to be read potentially by CD8 cytotoxic T cells. But because there are slight variations in the shapes of these MHC1s, oh, some of them, let's say, are the right shape to hang on to, the red dot, much better, right? Some of them are slightly the right shape to hang on to the green dot better, and some of them hang on to the blue dot better. Hopefully, you can already intuitively see why we evolved this way, right? Because the more different colored dots you can display at once that all came from different parts of bad clown, the greater chance of your immune system catching this, right, and seeing this. Okay. So in this case, all three of these are like the same full-fledged, privileged Hawaii driver's license. They're just, different patterns on the Hawaii driver's license, right? So these are all MHC1. They would all be interpreted the same way, but each one holds a different piece of bad clown a little better, right? So these proteins are highly polymorphic. So I should say, up until now, I've just been sort of casually referring to these as MHC platforms, right? Because I wanted to emphasize to you how they function, right? When I say they're like a little platform that something gets loaded on and displayed, it's easy for you to imagine that. But let's get a little bit more complicated here, because the reality is they are made, right, of these different little protein components. So we already said that the MHC1 types have one long protein and a small beta microglobulin. The MHC2 types are made up of two long strands of these proteins. These individual proteins, of course, are coded for by your genes, right? And you have different variations of these genes that are very similar to each other that help you code for 6 different types of MHC1 platforms on the surface of your cell at the same time, right? So every cell in your body that expresses MHC1, you have 6 different variations of MHC1, all being used at the same time on their surface, each presenting a different colored dot that came from bad clown to increase your ability for your immune system to see that there's something going on there, right? So these individual proteins that make up the MHC platform, we call them HLA proteins. So they were originally called human leukocyte antigens, right? It's kind of an antiquated name, and it's not a great name now that we know so much about how the immune system works, because you're saying, wait a minute, these themselves are antigens? Wait, what? No, but if you back up a long time ago, when immunologists were first beginning to understand how it is that cells like white blood cells, like lymphocytes, like T cells, can actually get excited and activate by pressing up against another cell, right? They identified, oh, it has something to do with these proteins on the surface of the other cell, right? Somehow communicating, showing something to the lymphocyte to get it excited. So they considered those like antigens themselves, right? They didn't realize that they carry around little peptides on them. So that's why they were called antigens. And because they were talking about white blood cells here, right, like lymphocytes, they called them human leukocyte antigens. And the name sticks, right? So that's why to this day they're called HLA proteins. So you can see we have 6 different HLA proteins that make our MHC class 1. So they are HLA A, B, C, E, F, G. Okay. Then you say, well, wait a minute, where's the HLA-D? It skips over because the HLA-D gene is the one that makes the chains that make MHC class 2. All right. So where are these genes located? They're located on chromosome 6, and this is their sort of map of how they're laid out. So you have a cluster, right, of the HLA-A, B, C, E, F, G genes that are together in this class 1 region, right? So that when they get transcribed and translated, the proteins that they make our 6 different MHC class 1 types. Then you have a group of the HLA-D genes, and this gets a little more complicated, right? Because they're very closely related, and so they're all called HLA-D, but you can see that they're called things like HLA-D-P, OA, M, OB, Q, and R, right? And so together, depending upon which ones are being expressed, you end up with 6 different types of MHC, one on your cells, and those particular special immune cells that have MHC2 on their surface, again, macrophages, dendritic cells, B cells, they also have MHC class 2 on their surface. When you look at this big cluster, you have these 2 clusters of genes. It's a big complex of genes, right? So when researchers first realized, right, that this complex of genes contained all the genes that were responsible for why when you transplant different organ tissues from a donor to a patient, right, you'd get rejection, right? Because at first when people were transplanting tissues, they'd be like, I don't get it, right? This is a healthy kidney. It came from a totally healthy person. This is a great healthy kidney. How come the patient's body is rejecting this kidney, right? They began to realize, oh, it has something to do with this complex of genes on chromosome 6. So that's why, because this is the major complex that influences that tissue rejection, they called it the major histocompatibility complex. And that's why the name MHC comes from that. And to this day, we call these MHC platforms, right? So get that straight, right? MHC platforms are made of HLA proteins, right? All right. These are highly polymorphic. So what do I mean by highly polymorphic? I mean, any gene in the human body, if you look across the population of all different people, you have variations, right? These variations, sometimes don't seem like they would do anything at all, right? In which case they just get carried through the population. Sometimes they might have an influence, right? They might actually have some influence on developing certain types of diseases, or they might have certain influences on other traits that aren't diseases, but just different phenotypic qualities that we have, right? I would say the majority of important proteins in our body are not super highly polymorphic, right? And that kind of makes sense because after eons of evolution, to get just that particular genetic sequence right so that the protein that's made from it is the right shape to perform a critical function, you would think that any sort of changes in that radically mess that up would probably not be evolutionarily favorable, right? But certain genes in our body are highly polymorphic. And in fact, these HLA genes are the most highly polymorphic genes in the human genome. Right? Some of these HLA types, like HLA-A and B and C, there are thousands of documented, cataloged human variations, right, that occur in these genes that are passed on due to your ancestry, right? So you inherit these polymorphisms depending upon all of your ancestors before you, what polymorphisms they had, they get passed on, right, in your family lineage. Why is this significant? If you look here, the color coding gives you an indication that HLA-A, B, and C genes are the most highly polymorphic, Whereas some of the other ones, like, I don't know, HLA-E here is kind of oligomorphic, meaning there's more like a handful of different polymorphism. Some of them are more stable, like the yellow ones, like HLA-F and G are monomorphic, right? So in this case, there are no variations in the gene sequence from different people, right? So how does this tie in with what I've been discussing so far about how your cell wants to display different bits and pieces, as many different pieces as possible of peptides from something like a pathogen? So that's where this cartoon comes in. I love this cartoon. I think it's funny, but it gets a really good point across, right? Your makeup, your genetic makeup right now, right, means that you have a combination of different polymorphisms that you inherited from your parents, your grandparents, great-great-parents, all the way back, right, your family tree, right? These certain polymorphisms in these HLA genes were passed down to you, and now in your body, these are the versions of these HLA, A, B, and C genes that you have, right? So the proteins that are made by them, your HLA A, B, and C proteins that make MHC1, right, reflect slightly different shapes that are based upon these polymorphisms that you inherited, right? So let's look at this example here. This is from Trends in Immunology, which is a highly respected journal in the field. So let's pretend inside of a cell you have a protein, right? So this chicken here represents, a protein that's going to get broken down by the proteasome and recycled, right? I don't know why the proteasome is represented by this red guy with a saw, and for that matter, I don't even know why he's wearing a top hat, but okay, let's go with it, right? So the proteasome breaks down the peptide into little pieces, right? Chicken head, chicken wing, chicken leg, chicken foot, chicken, I don't know what this is, chicken butt. I don't know what the brown thing is coming out of the butt, but okay, right? So these all have a chance of being presented on the outside of this cell if the cell has the right shaped polymorphisms, the right shaped MHC1s to be able to properly hang on to these and display them. So let's look. What this is saying here is based upon the ancestry of this individual whose cell this is. Maybe they have some Asian ancestry in them, right? And so the polymorphisms that are in their HLA-A gene came from Asia a long time ago. Maybe they have some polymorphisms that they carry from a little mixing of European, right? This is Mediterranean, maybe ancestry, like Greek and Roman. So maybe there's a polymorphism in their HLA-B gene that reflects they have Mediterranean ancestry. And this is maybe European, right? I assume it's European because it's like their raincoats with umbrellas. I don't know why that means anything, but anyhow, so they have polymorphisms due to their European ancestry all mixed in. So as a result, because of that, these examples of MHC1s on this person's cells, right, gives them the ability to preferentially be able to display chicken head on HLA-A, chicken wing on HLA-B, chicken foot on HLA-C. Although I always say they should have made the Chinese guy carrying around a plate with chicken feet on it, but whatever, right? Why is that important? Because if you have a circulating T cell that's coming around, this T cell, its T cell receptor will only recognize, right, a certain type of peptide here. So if this T cell's T cell receptor by chance is the right shape to pick up on chicken foot, right, then this T cell has an opportunity to recognize this over here and start an immune reaction, right? Hey, it detects chicken foot. That means this chicken pathogen protein was present. But what if this individual, this person, didn't have the polymorphism that comes from this, European ancestry? So therefore, even though the cell on its surface could display chicken head, chicken drumstick, chicken wing, chicken butt, it doesn't display chicken foot very well. Then this T cell would miss this, right? So I hope you see now. That's why evolutionarily our HLA genes are so polymorphic, because the more rich mixture of different possible polymorphisms you have, the slightly different shaped MHCs that you have, the better chance that you can display all parts of the chicken so that T cells can see it. Right? So this means in this person's case, not only could this T cell that recognized chicken foot get activated, but if he also has a T cell that recognizes chicken head and a T cell somewhere that recognizes chicken drumstick, those could also activate. And that would be a stronger, more effective immune response that started. Right? Okay. So this goes to show you, I don't know who the author was of this, I've forgotten, but I think it must have been really a famous, highly respected author, because once you've made it in your career, you can publish cartoons like this in highly respected journals. All right, I can't do that. All right, anyhow, so HLA polymorphisms strengthen our immunity, right? So if you look at here, if you look at MHC class 1 genes, so the HLA-A, B, and C are some of the most highly polymorphic genes. So these are all the cataloged different polymorphisms that have been identified in the human population, right? Thousands of them. Whereas things like MHC class 2, maybe it's a little bit more limited. I mean, some of them, like the HLA-D RB1, you have a lot of different cataloged polymorphisms, right? But some of the other ones, there's only like a handful of variations. Right? How do we see evidence that this is important? We see that's important in studies like this. So this is a clinical trial where they were looking at people who were HIV positive, right? And when they were in the clinic, they had some blood samples taken so they can determine what sets of polymorphisms do they have that they inherited from their ancestry, right? And in the case of this study, they just broke it into 3 big groups, right? If you had a certain set of polymorphisms, they put you in the red group, right? Another group of polymorphisms from your ancestry, they put you in the yellow group. Another set, they put you in the blue group, right? And they followed these people over time. So over many years since they were first diagnosed with HIV, how long did it take before their immune system finally could not keep up with the HIV and these people developed full-blown AIDS, right? And what do you see here? Very clearly, depending on what set of polymorphisms you have, right, your prognosis in terms of your ability to fend off the development of AIDS as a result of HIV infection, right? It's very clear. So what does that mean? It implies that people with the red set of polymorphisms that they inherited, right, from their ancestors, for some reason, their immune systems are better at keeping the HIV under control. Why? Because they have the particular MHC1s that do a better job of showing a diverse range of small peptides that come from the HIV virus on the surface of their infected cells. So those cells then, advertising that they're infected by HIV, right, there's greater opportunity for their T cells to come and see that and quickly tell that cell to die before virus particles get spread further. Compared to the people in the blue group, right, maybe due to their polymorphisms, right, they're not displaying all parts of the HIV or examples of peptides from HIV. So it's like saying, right, with the chicken, they're not displaying the chicken foot or the chicken head. Maybe they only display the chicken wing. And so therefore, only T cells in their body that are designed to recognize chicken wing have an opportunity to respond, right? And their immune system misses out on an opportunity to be able to quickly identify this target. So what's fascinating about that is what it means is that these HLA polymorphisms are good for us as a species, right? Because you yourself, the moment you were conceived, that determined your set of polymorphisms, right? That doesn't change throughout your life. So you are either in the blue group, the yellow group, or the red group here. So for you, if you luck out, you're in the red group, right? If you're in the blue group, too bad for you, right? But maybe being in the blue group is actually better for other types of pathogens, right? So that's why as a species, this richness of HLA polymorphisms persists through evolution, right? Because it keeps our species, gives us a fighting chance to adapt to new threats that come in and threaten us with extinction in the form of pathogens. All right, last slide. Sorry, I'm going over. The last thing I want to touch upon is how this is relevant to this whole discussion of major histocompatibility, right? Remember, I told you these things are called human leukocyte antigens, right? The proteins that make up these MHCs, because the MHC is the big group of genes that make these HLA proteins, right? And why are they called MHCs? Because originally researchers saw that these genes were related to the rejection of tissue. So what's going on here? Let's imagine in your body, right? Here's a typical body cell in your body, and you make your HLA type, right? Because you were born with whatever polymorphisms you got from your mom and dad to make this particular shaped yellow HLA-A. So normally your body cell, A, it's happily just displaying normal peptides that are coming from inside of it on HLA-A on this MHC1 platform, right? So that's you. The peptide is you being displayed on MHC1. Therefore, you're not going to have T cells that are the right shape to detect this. Right? We will learn about this after the first exam, how it is that you actually determine what T cells and B cells, for that matter, are allowed to go around in your immune system. But clearly, there's a selection process here, right? Because the only T cells that are allowed to be in your system are ones that do not recognize this combination of shapes. Therefore, this T cell would leave this cell alone, right? It's different if you're infected by bad clown, right? Because your HLA-A type, oh, it's a good one for carrying and displaying this part of bad clown on it, right? So that's bad clown's identity being displayed on your MHC1 type. So your T cells, there will be one, hopefully, that is the right shape, right? Through VDJ recombination, it stumbled upon the right shape to detect this different shaped peptide that's foreign in the context of MHC1. All right, so that so far, I think, fits your understanding of how this works, right? So the question is, what if instead of your normal body's tissue, right, you're dealing with somebody else's tissue that's put in your body? In other words, a transplantation, liver transplantation, lung transplantation, kidney transplantation. What would that be like? Well, it comes from a different person. Therefore, their HLA polymorphisms are different than yours, right? Because theirs is the green type that came from their mom and dad. So the problem is, even though there's no infection going on, right? Let's say this is a kidney cell. The kidney cell is still presenting normal, healthy peptides coming from in it. So it still is you, right? But the problem is it's displaying it on somebody else's MHC type, right? So this shape, this combined shape, is foreign to your body. So that means there will be another T cell in your body that by chance is the right shape to bind this strange looking foreign combination. So that is like your identity being presented on the wrong type of driver's license, right? And as a result, your body will react against this as a foreign antigen, right? And the asterisk here is just to say that the specific TCR here, this bright purple one, rejects the donated tissue cell because it's allowed to exist in your body because it never would have naturally encountered this green HLA, their type, previously in your body. So there was never any incentive, right, to prevent this T cell from working as part of your immune system. Okay. All right. Let's go ahead and stop there.